Let's move on to clinical assessment during this webcast. We'll talk about clinical assessment in two different areas: energy metabolism assessment and standard clinical assessment. And let's begin with assessing energy metabolism, or with the energy metabolism assessment that we utilize clinically. Naturally, if we subscribe to the idea, the theory that impaired energy metabolism is this common link in chronic degenerative type pathologies, and we haven't identified very many of those conditions, chronic conditions that don't pertain to this methodology, then it really behooves us to employ a clinical model that's actually geared toward energy metabolism. And that's really the first pillar of a two-pillar assessment model, the second pillar being the standard clinical assessments, and we'll discuss those as well. But this first pillar is extremely important to understand. Again, the second pillar is just standard clinical assessment that refers to the mainstream diagnostics, which are very effective. I mean, you can accuse the mainstream of a lot of things, but when it comes to diagnostics, at least with respect to correlative variables or signs of an underlying pathology, they really shine in that area. Okay, there's no need to invent the wheel there. But I really want to focus on Delta's clinical method pertaining to assessing energy metabolism during this webcast. And we'll discuss that in four different areas: hydroperoxide spectrophotometry, where we're actually taking a look at the hydroperoxides, the role of hydrogen peroxide in both the cause but also in the resolution of these type of pathologies. Plasma antioxidant spectrophotometry, we'll take a look at that. Open respirometry and biophoton absorption assessment, but the main thing we're going to be spending time on here is the hydroperoxide analysis. This is really where everything starts to come together from the previous webcast. This is where we begin to connect the dots, if you will, between the theory of causation, the mechanisms of impairment, and the assessment model that really allows us to quantify the various parameters involved. Again, if we're dealing with impaired energy metabolism as the basis for a given condition, then we should be able to utilize some clinical measure or measures that pertain to impaired metabolism. And the first of those measures is hydroperoxide analysis. Normal values for hydroperoxides as a healthy individual range from anywhere between 250 and 300. And clinically, we want to identify a baseline. That's the first thing that we're looking at. And for people with chronic conditions, it ranges anywhere from about 320 to 500. And in a previous webcast, we discussed that in most chronic pathologies where energy metabolism is impaired, we're going to see an increase in free radical formation in oxygen metabolites. We call them reactive oxygen metabolites, or if you want to call them reactive oxygen species, ROS, right? So we view this increase in ROS as a protective response. Remember, this is a dysfunctional protective response where the cell's generating reactive forms of oxygen in an attempt to deal with an underlying pathogen, in most cases the stealth pathogen or filterable forms, but the response is dysfunctional. Or we could say that the peroxides produced are dysfunctional, as outlined in the last webcast. You'll remember that the mitochondrial produced peroxides also had defects in polymerization, reactivity, oxygen reduction potentials. They're not able to get the job done, right? So why are we so interested in this hydroperoxide analysis? So we can determine a move from moderate levels of hydrogen peroxide to higher levels. And we're going to explore that in greater detail here in a minute. But remember, in order for H2O2 to function as a signaling agent, its concentration must increase above a certain threshold. And moderate levels do nothing but damage the cell. Now, that may seem odd at first glance. It sounds like we're saying that we want to increase the oxidative stress of the cell. And to some extent, that's true. But an increase in what and why? Well, let's tackle the what, or let's tackle the why, and then we'll tackle the what. Because hydrogen peroxide plays a critical role as a signaling molecule in the regulation of mitochondrial respiration. There are a host of of signaling pathways that are actually controlled by H2O2 that regulate the entire disease mechanism, these pathways. That's the first reason. The other is due to its antimicrobial action within the mitochondrial membrane. So when we say we want to trigger an increase in H2O production, yes, but it's also important to understand that we're seeing this corresponding decrease in superoxide and hydroxyl radicals. This probably has something to do, we suspect it has something to do with the restoration of a pathway that signals the shutdown of oxygen radicals and other parts of the membrane, but we're not sure about that yet. But the practical achievement that we're trying to identify there is just an increase in the good radicals and a decrease in the bad radicals. Keeping in mind that there are a couple of parameters that we really should probably qualify here. Number one, we're looking for an increase specifically in high ORP value peroxides. These are peroxides that pack a punch. They're not the same simple peroxides that are involved in oxidative stress in general. These are super peroxides, okay? Number two, we're looking for an increase in hydroperoxide formation within a specific part of the membrane, okay? This, in general sense, that would be the complex 1 protein. In more specific terms, that would be the semiquinone pathway. Again, the importance of honing in on the zone associated with the mechanism of impairment and triggering an increase in superperoxides that occur catalytically at the site of impairment. You may recall from a previous webcast that under normal circumstances, when fuel hits the mitochondria as NADH, the hydrogen is stripped off, and among other things, a free radical, namely semiquinone, is generated, right? And as long as there's an ample supply of molecular oxygen, that free radical eventually forms hydrogen peroxide that actually burns, we discussed some of the roles, but it burns up pathogens and sustains signaling pathways and so forth, and prevents the microbes, filterable forms, their unsaturated proteins, etc., from disrupting energy production. But in the absence of oxygen, under hypoxic conditions, the semiquinone radical actually adds to the NAD plus and blocks the addition of hydrogen to form NADH. So the wheels of energy production at that point just come to a screeching halt. And there's nothing novel about moderate ROS production, moderate reactive oxygen species production, in response to a chronic condition. That's what we generally refer to as oxidative stress, and that's widely accepted as a hallmark feature in chronic pathologies, increased ROS. But what they won't necessarily concur with is that it's an attempted protective response. And understanding that has been indispensable for us in understanding the potential of the peroxide to correct the metabolic disorder. If we can just help the system along, help it achieve what it's attempting to do. So what is it clinically that we're trying to measure with this device, this FROS4, this Italian photometer? The downstream regulation, or the downstream restoration, if you will, of H2O2 production. And we're looking specifically at that within the semiquinone pathway because we need that increase in H2O production, H2O2 production to occur specifically within that pathway. Because the mitochondrion wants to produce H2O2 specifically within that pathway, but that's where the impairment resides, so it's not able to get the peroxide production going in that specific area. Most of the H2O2 that we're measuring initially is just coming from different parts of the membrane, from the complex 3 protein in particular. So all we're really doing clinically is we're just confirming that we see that we're able to measure an overall increase in peroxide production. But what we're measuring with this device, this FROS4, there's another interesting aspect to that. This technology is generally used to assess oxidative stress. Most of the practitioners using the device would do so with the intent of seeing a decrease, okay? A decrease, not an increase, in the level of hydroperoxides. And, we can appreciate that because if you can't peak the peroxide properly in order to achieve signal propagation, then the next best thing to do is to bring it down. And they do that generally by quenching free radical production. Our approach is fundamentally different, and one that of necessity recognizes the role of antioxidants in the equation. So if you look at some of the traditional models, prevailing integrative type therapies, they're generally focused on increasing antioxidants. And why do they want to do that? Because they want to buffer free radical activity. The basic idea is bring down the oxidative stress by increasing antioxidants. And in a world without the BX, I would agree that this is a somewhat viable approach to at least decreasing the free radical damage, reducing oxidative stress. But quenching free radical formation, that's an approach that subscribes to the idea that homeostasis or balance in biological design is always a good thing. We have to have this balance between free radicals and antioxidants, right? But my question is balance really always a good thing? Because most will argue that excessive reactive oxygen species formation can induce oxidative stress. True. And that oxidative stress leads to cell damage. Again, true. So naturally, when we look at antioxidant function in terms of its scavenging function, scavenging excessively produced reactive oxygen species, buffering free radical formation, right? We think of that as a good thing. And more importantly, that it's always a good thing, because homeostasis, after all, is in fact a good thing, right? And I'm not arguing with, that concept as a general rule. But have you ever stopped to ask yourself why cells tend to shift towards the formation of free radicals in the first place? Cells tend to be slightly imbalanced in this regard, right? We propose that the cell is attempting to achieve a differential between the antioxidant and the free radical. And here's how we came to that conclusion. Our studies, through the use of hydroperoxide analysis, through the use of the FROS4, show excessive antioxidant values anytime we have excessive levels of hydroperoxides. So high oxidative stress equals high endogenous antioxidant levels. These are antioxidants originating from within the mitochondria. Okay? And from the perspective of homeostasis, that makes perfect sense. But here's where it gets interesting. As we add the BX into the equation, we're going to see this increased hydroperoxide formation. That's what we were wanting to see. And we're going to see a decrease, not an increase, but a decrease in antioxidant formation, which is kind of a problem because the FROS4 is intended to measure the exact opposite. They're looking for increased antioxidant production, decreased oxidative stress, and we have this inverted objective. So Mike, our data analyst, came up with what we call a relative score, which basically says that any decrease in antioxidants represents an increase in hydroperoxides. So in order to measure how high the peroxide peak actually was, we had to say that a 1-point reduction in antioxidants represented a 1-point increase in peroxides, because naturally the antioxidant that is consumed in the process of canceling out a peroxide has to be accounted for, right? And clinically, here's what we see. A decrease in the antioxidant value along with an increase in hydroperoxides, and a relative increase of 20 to 30%, 20 to 35% is really all it takes. But that decrease in antioxidants with the increase in hydroperoxides corresponds with an improvement in almost every measure of the condition. In other words, under normal conditions, as oxidative stress increases within the moderate range, and this could be cancer, Lyme, Parkinson's, cardiovascular, whatever, but as that oxidative stress increases within the moderate range, we'll see an increase in symptoms. We'll see an increase in biomarkers reflecting the disease process. But with the BX, we see just the opposite. With a 20 to 30% increase in the superperoxides, this is the relative score, we see an average of 36% decrease, for example, in the erythrocyte sedimentation rate across all conditions, and a 42% decrease in C-reactive protein, both of which are inflammatory markers, inflammatory measures, right? So how do you explain that? 20-30% increase in the superperoxides, and yet we're seeing 30-40% decrease in all the inflammatory markers. Not only that, but we'll see improvements in VO2, VCO2, and the exchange ratios as measured in open respirometry, and we'll discuss that in a bit. We see an average 13% improvement in blood pressure, where high blood pressure is actually part of the equation. And we'll explore specific case studies in the upcoming webcast, but suffice it to say that we generally see improvements across the board. And we're talking about standard diagnostic standard biomarkers of any particular condition across the board. If it's rheumatoid arthritis, see changes in RA factor. If it's cardiovascular, we'll see changes in cholesterol, triglycerides, blood pressure. If it's diabetic conditions, we'll see major changes in circulation, sugar levels, etc. And these are all correlating to improvements using standard clinical assessment, regardless of the condition, as long as the condition has a correlative relationship to oxidative stress, which most of the chronic conditions do. And we'll explore some specific numbers on that during the case studies webcast, the upcoming webcast. And of course, I'm discussing these changes in the context of our Asian operations. If you're a doctor in a jurisdiction that's controlled by the drug cartel, then you may or may not be able to have this frank of a discussion, as it may cross the boundaries of general intended use, just injecting a disclosure there. But returning to the measures directly corresponding to a peak in what we call high order peroxides or super peroxides, we'll see a decrease in joint pain. We'll see improvements in GI function, in Lyme and in Parkinson's. We typically see major improvements in cognitive function, improvements in gait, vestibular function, energy, endurance, athletic performance, etc. All of which tend to be impaired with anyone presenting with moderate levels of oxidative stress. And we see these improvements when the intended protective role of the peroxide is achieved. That's what we typically see. I think it's also important to state that we'll generally see the peroxide levels peak, and as recovery commences, gradually over the course of a year or so, the peroxide levels will begin to decrease until such time as the system kind of moves back toward a balanced redox system, with the antioxidants and the free radicals returning to normal homeostasis. So I didn't want to somehow imply that homeostasis is not important. I didn't want you to get that impression, but there is such a thing as what we call therapeutic disequilibrium. Okay? And it's really through this type of clinical assessment, the FROS4, that we've been able to demonstrate, that in order to achieve balance, we first have to create an imbalance in the system. So perhaps that's why James Watson, who won the Nobel Prize for his work in identifying the double helix structure of DNA, recently stated that, and I quote, "The fundamental cause of inflammatory conditions is a lack of biological oxidants, not an excess," end quote. Pretty celebrated geneticist here, and he's saying that antioxidants are potentially the culprit. And it's a bit more complex than that, because even if the biological design were such that the system inherently moved toward a temporary imbalance with respect to the peroxide formation, the peroxides formed still have to be a high order or a high ORP peroxide produced within the correct pathway. Well, the BX solves many of these problems. I know we're running short on time, so let's move on to the other two areas of clinical assessment that we use, specifically, again, to look at improvements in the energy-producing mechanism of the cell. One of these is what I call a crenation test, or a membrane permeability test. It's conducted under a slide, under a microscope. You could consider it a form of live blood analysis, but the thing I like about it is it has this quantifiable aspect to it that allows us to specifically identify the normalization of energy metabolism. So if we take a blood sample and we subject it to a 1% molar solution of sodium chloride under the microscope, okay, normal erythrocytes will shrivel up because the 1% salt solution is hypertonic to the 0.85% normal osmotic pressure inside of the red blood cell. But a sick cell with impaired energy metabolism will refuse to shrivel up. You're going to likely see it swell up because the osmotic pressure inside of the cell is actually elevated beyond the 1%. Okay? Why? Because the sodium-potassium pump, and particularly the specialized proteins in that pump, consume about 40% of all the ATP that's produced in that cell. And this lack of energy production, it basically causes the protein structures within the membrane to kind of fragment, okay? They just collapse on themselves in a manner that shuts down the exchange across that membrane. Under optimal conditions, healthy individual, we should see about 97 to 100% crenation. That's what we typically see. And as we videotape that field, we do it during a 60-second exposure, then we go back and we're actually able to grid the cells and count it and throw it on a spreadsheet. Again, we're simply counting the number of cells that shrivel up in a 1% sodium chloride solution. And with most of the chronic conditions, prior to beginning the BX, we'll see anywhere from 40 to 70% crenation. But after the BX, 2, 3, 4 weeks after the BX, we'll see right around 100% crenation in about 90% of the cases. Even though we have to emphasize that health restoration, okay, truly getting to the finish line is about a one-year proposition. Because you have to remember, recovery follows the cell cycle. The crenation test is a test that's conducted on the red blood cells. If you look at the typical mature erythrocyte lives for about 120 days in the circulation, which is a relatively short life cycle. So naturally, we're going to see a restoration of oxygen carrying capacity. We're going to see an improvement in the general function of the red blood cells long before we're going to see any kind of restoration in the neurons, for instance. If we understand that the restoration is tied to the renewed genetic material that will not fully manifest until sometime after cell division, where the daughter cells are equipped with this corrected machinery. In summary, what we should see is a complete alteration in the responsiveness of the cell membrane. That's a direct result of restored energy metabolism. Though erythrocytes they're not equipped with mitochondria, they still produce energy via fermentation. And they really do offer a qualitative measure that provides a very strong correlation to mitochondrial respiration in general. Another clinical assessment tool that we use is open respirometry, or what we refer to as open flow respirometry. It's something you'll typically see used in athletic performance testing or exercise physiology, where they throw you on a treadmill or a bicycle or an elliptical, and they have you breathe into a mask that simply collects the gas output for analysis. In the context of chronic pathologies, oxygen consumption and CO2 production are a very good measure of energy metabolism in general. So a tool like this becomes very useful. We use core technologies. I think it's the Cardio Coach, but there are a lot of units out there that work just fine. The basic idea is any chronic disease process with hallmark features, with impaired metabolism, but also with oxidative stress involved, those types of conditions will generally present with major alterations in O2 utilization or O2 consumption. We're not talking an exercise response here. when we're measuring improvements, because these tests are performed at rest. So generally what we'll see prior to the BX is we'll see compromised volumes, we'll see compromised exchange ratios between O2 and CO2, basically a clear indication that we just don't have efficient energy metabolism going on. After the BX, we'll see the exchange ratios move closer to 1.0, and the exchange ratios should ideally be 1-to-1, be a 1-to-1 ratio with respect to O2, CO2. We tend to see less O2 after the BX, less O2 in the expired gases, because naturally we're seeing O2 utilization increase overall, so we're going to see less expired O2, and we'll see vast improvements in total energy expenditures, total energy output as well. So overall, just a very effective clinical tool to assess mitochondrial function. And again, we'll dedicate a webcast to the use of photon absorption assessment in the near future, so stay tuned for that, and we'll explore some fun things there as we're building an assessment diagnostic tool in that area. But thank you for joining. Make sure to stay tuned for the next webcast entitled Case Studies, where we'll explore some very specific numbers pertaining to symptomology, blood work, etc. Okay? Talk to you soon.